Now, organization of nerve. Organization of nerve is nothing but coverings of nerve. Many nerve fibers. Now, many nerve fibers together is known as fasciculus. Now, many fasciculi together is known as a nerve. That is how a nerve is formed. Now, this nerve contains three coverings. First one is endoneurium, second is perineurium, and third one is epineurium. Now, what is endoneurium? It covers each single axon. It covers each single axon. Whereas perineurium, it covers each bundle, which means each fasciculus. Many nerve fibers together is known as a fasciculus, right? So, each fasciculus is covered by a perineurium. Now, epineurium covers the whole nerve, which is many fasciculi together, they form one nerve. That nerve is covered by epineurium. Okay, whole nerve is covered by epineurium. Each bundle is covered by perineurium. Now, this is the cross section image of a nerve. Now, you can see these are the blood vessels. This is the epineurium. Epineurium is the covering of the whole nerve. Covering of the whole nerve. Okay. Perineurium. What is perineurium? Bundle. We just spoke about it, no? Bundle of nerve fibers. See? Covers each bundle, which is fasciculus. So that is perineurium. So this is a perineurium. This is a whole fascicle. Now this is a whole fascicle and the covering is known as perineurium. Endoneurium is only the each single axon. So this is each single axon is covered by endoneurium. These are the nerve fibers which are present. Now did you understand? Epineurium, whole nerve, perineurium, fasciculus. This is the one fascicle here. This is one fascicle here covering to that fasciculus. And these are the nerve fibers present inside the fascicle. Endoneurium is covering one single axon. Now nerve fibers, types of nerve fibers, you can see there are two types of nerve fibers. First one is myelinated and the other one is non-myelinated. Now myelinated axon is nothing but axon which is covered with myelin sheath. Now with myelin sheath, the signals are sent fast. So the faster the signals travel and it is in white color. Non-myelinated is nothing but it does not contain any myelin sheath. Hence, the signals are slow passages. So the passage of the signal is slow. Now key point here is proteins are made in cell body by nasal bodies, that we know. Then they are sent down by the axon by the axonal flow because axon can't make its own. That is also a known thing because it does not contain nasal bodies. It is sent down to the axon because it doesn't have its own. Now myelinated and unmyelinated nerve fibers. Okay, this is the axon here. This is the unmyelinated nerve fiber. It contains the cell body which also has a nucleus and these are the dendrites. Now this is the myelin sheath that is the Schwann cell. It contains a Schwann cell, and this is hence it has myelin sheath. It is known as a myelinated nerve fiber. Now this is myelinated and unmyelinated nerve fibers. Both types use Schwann cells, but in a different way. Okay. In myelinated nerve fiber, which is the insulated wire, which has an insulated wire. So myelin sheath, which is the fatty white layer wrapped around, this is the myelin sheath, fatty white layer wrapped around the axon, this is the axon, wrapped around the axon like a tape. It is wrapped around the axon like a tape. Okay. So what does this do? This myelin sheath does. What will this myelin sheath do here? It will help the signals move fast, like jumping between the gaps. Okay. So it will help it to move fast. The outermost layer of the Schwann cell is known as the Neurilemma. This one. This is the outermost layer of the Schwann cell. So the Schwann cell, this is known as the Neurilemma. This is the nucleus of the Schwann cell. Okay. And why this Neurilemma is important? Because whenever the nerve gets cut or anything for the repair, the Neurilemma is important as it helps for the repairing of the nerve. Okay. It helps in the repairing of the nerve. Okay, next is unmyelinated nerve fibers, where bare wires in one sleeve. These are all the wires in only one sleeve. Okay. No myelin sheath is present here. This is the axon. No myelin sheath present here. Hence the signals are travelled slow. It is made up of one Schwann cell which just holds many axons in its cytoplasm. This is only one Schwann cell which holds many axons. This is one axon, this is one axon, this is one axon. So it has many axons in its cytoplasm. This is the nucleus of Schwann cell. So the cytoplasm of the Schwann cell has many axons present in it. So Neurilemma is still present here. If you can see this, Neurilemma is still present here. It's the outer part of the Schwann cell. It is the outer part of the Schwann cell. So it is basically found in nerves that don't need any speed. As it travels slow, in nerves that don't need much speed, like in organ functioning and all, it is little normal and in a normal pace way. Okay, that is unmyelinated nerve fibers for you. Next is myelin sheath. Now the myelin sheath is nothing but, we just saw here myelinated things. Now you can think about it in further. Insulation around the nerve fiber. As you have seen here, insulation around the nerve fiber. It is thick fatty protein cover around the axon. You can see around the axon. Then color makes nerve fibers look white. It will make nerve fibers look white because of the fatty protein cover. It is not continuous. See, myelin sheath is not continuous. It has gaps, and that gaps is known as nodes of Ranvier. Those gaps are known as nodes of Ranvier. Okay. So the internode is nothing but part between two nodes is known as an internode. So what is it made up of? It is made up of, myelin sheath is made up of lipoproteins. Lipoproteins and fats. So what is lipoprotein as such? Lipoprotein is nothing but fat plus protein layers stacked like an onion. Now, if you can see, isn't it stacked like an onion? the same way. Myelin sheath. It is stacked like an onion. Fat and protein layers. Now what are fats here present? Cholesterol, lecithin, cerebroside. These are few of the fats which are present here. Now how it is formed? It is formed by myelinogenesis. The formation of the myelin sheath is known as myelinogenesis. Now it is made up of Schwann cells in peripheral nerves. Now you know Schwann cells, we just spoke about it. Now what will this Schwann cell do? It wraps around the axon many times like a tape. We have seen here, it wraps around the axon many times like a tape. Okay, now layers fuse to form myelin. Now these layers fuse to form what? Myelin. So cytoplasm gets squeezed out completely. Okay, outer part of the Schwann cell stays as Neurilemma. We have also seen the outer part of the Schwann cell, which is Neurilemma. So nucleus sits between myelin and Neurilemma. Now what is happening? As it is all completely like a tape, the nucleus is squeezed outside. Can you see the nucleus? It is actually squeezed outside, and the nucleus of Schwann cell sits between Neurilemma. Myelin and the Neurilemma. Now this is the myelin sheath, right? Between one myelin and the outermost layer, which is Neurilemma. So the nucleus of Schwann cell sits between the myelin and Neurilemma. Now nucleus sits between myelin and Neurilemma. So what is the timing? It starts around 4th month in the womb and finishes up to 2 years old. Now this is the image of the myelin sheath. Okay, blue coating label, that is the myelin sheath, the blue coated label here. This is the myelin sheath. Okay, fatty layers wrapped around the axon like a tape. We have just sawn now. Then what is happening here? Think of it like a rubber insulation, okay, around the wire. So what is happening here? How does it works? Myelin lets the signal jump. So the myelin sheath, it helps in the signal jump instead of crawl. Now crawling is nothing but moving slow. The signals are moving slow like a crawl. Now this myelin sheath will help instead of crawling so that it can jump and move fast. The signals can move fast with the help of the myelin sheath. Now what is the node of Ranvier here? These are the nodes of Ranvier. What does they do? The red gaps between two myelin pieces is known as the nodes of Ranvier. What is its job? The bare spots where signal jumps to next section. From this section to this section. You can see from here it literally jumps. Then here it literally jumps. Like that's how it moves fast. The signals are moving fast. Okay. Like skipping the stones across water. That is how the jump takes place. Now inside the axon, this is the axon, right? So inside the axon you can see microfilament and microtubule. Okay. These are nothing but the microfilament is not like a microtubule is like a train and microfilaments are like a train track. Supporting beams. This is like a track and this is like a supporting beam. Okay, inside the axon they help to move fast the stuff and keep in shape. So the filaments help in keeping in shape and the microtubule help in moving fast so that the information, the signals are moved fast with the help of the microtubule, and microfilaments are the supporting beams. So in one line, what can we say? Myelin sheath is nothing but a fatty wrap made up of oligodendrocytes in brain and spinal cord. What will it do? It is insulated in the axon, so the signals can jump fast from one node of Ranvier to the next node of Ranvier. That is how it moves fast. Okay. Now next, two main functions. Two main functions of the myelin sheath. First one is it makes signals faster, as we just saw. Impulse jumps from one node of Ranvier to the next one, called as saltatory condition, like jumping stones. Okay, without myelin you can see slow. Signals are passed slow. With myelin up to 100 times faster you can see the signals moving. Now it also acts like insulation. How? It stops electric signals from leaking out. Insulation is nothing but keeping it intact and not leaking out, right? So it stops the electric signals from leaking out. It prevents one nerve from accidentally exciting the neighbor. So one nerve, it has full of electric signals. So accidentally it shouldn't go and touch the leak out and excite the other nerve. Okay, that is how if there is myelin sheath, it gets insulated, so the electric impulses cannot move out and it cannot excite the adjacent nerves. So it is like a plastic coating on the electric wires. Now what is Neurilemma here? Thin outer covering of the nerve fiber. You remember we just saw here. So what is it? It is a thin membrane around the axon, also called as sheath of Schwann. Okay, where it is found? It is found in both non-myelinated nerve and myelinated nerve. In non-myelinated nerve, Neurilemma directly touches the axolemma, whereas in myelinated nerve, Neurilemma covers the myelin sheath. You remember? It is the outermost layer of the myelin sheath. Okay. And at the node of Ranvier, it dips in like a finger to touch the axolemma. Okay. How? Let us see now. Now can you see that this is the nucleus of Schwann cell? Okay. This is unmyelinated. This is myelinated nerve fiber. Here you can see Neurilemma. Okay. This is the myelin sheath. Now, outermost layer of the myelin sheath, you can see Neurilemma. It comes and it dips at nodes of Ranvier, and again it moves along the myelin sheath. So this dip we were talking about. Okay. See, at node of Ranvier, it dips in like a finger to touch the axolemma. This is the axolemma, right? So to touch the axolemma, it will dip a bit. At nodes of Ranvier, everything, all this has nodes of Ranvier. Between one and the other, between two myelin sheaths, you have nodes of Ranvier. And there you can see, this is node of Ranvier. There you can see a dip of the Neurilemma. And again it goes along the myelin sheath. Whereas in unmyelinated nerve fiber, it directly touches the axolemma. This is the axon and this is the axolemma. So it directly touches the axolemma. So Neurilemma touches the axolemma here. Okay. Now what are the functions here? In non-myelinated fibers, it just acts as a protective cover. Whereas in myelinated fibers, it is needed to form myelin sheath. Okay. Here, it just acts like a protective cover in unmyelinated nerve fibers. Whereas in myelinated, it helps in making the myelin sheath as well. Neurilemma. Okay. Why Neurilemma is important? Neurilemma is only in peripheral nerves. Okay. It is not in brain or spinal cord. Okay. There what is happening? Oligodendroglea are present, making myelin sheath there in brain and spinal cord. So why Neurilemma is important again? It helps in repairing the nerves. Also, when we have talked about the nerve functions and all, so the Neurilemma helps in repairing the nerve. Hence, as it is absent in brain and spinal cord nerves, That is why the CNS repair is slow. Okay. Whereas the other PNS peripheral nerves repair is faster compared to the brain and spinal cord nerve repairs. Now neutrophins. Neutrophins is nothing but nerve growth proteins. So what are they? Protein substances that make nerve grow. That makes the nerve grow, survive and work properly. So all of that protein is nothing but to grow, survive and work properly, right? So it is made up of muscles, astrocytes, which is nothing but it is made by muscles. It is made by muscles, astrocytes. Astrocytes are nothing but brain supporting cells and neurons. Okay. What are the four main jobs what neutrophins does are? It helps baby neurons grow. So it helps the baby neurons to grow. Baby neurons to grow in brain and body. Whereas it also helps in neurons to survive. That is what we also talked about. What are they is nothing but protein substances that help nerve grow, survive and work properly. So it helps baby neurons to grow. It helps neurons to survive and repair after damage. And it maintains healthy nervous tissue. Okay, helps signals pass between neurons. It helps neurons grow. It helps neurons survive and repair after damage. It maintains healthy nervous tissue and it also helps signals pass through between the neurons. Now, what is the cool fact here? Scientists found that they can help damaged neurons regrow in lab and animals. So we can also hope that it may help treat Parkinson's, Alzheimer's someday. Okay, already used in some medications, the formula, but still it is not yet confirmed. And how do they actually work here? Neutrophins, they stick to the receptors on the neurons and there they turn on the tyrosine kinase. And what will this tyrosine kinase do? This tyrosine kinase tells the cells to grow or repair. Okay. Now the first one discovered, the first neurotrophins, the first neurotrophin which is discovered is nerve growth factor. Okay, now many other types have come, but the first one was nerve growth factor. Now we'll be talking about the nerve growth factor. Nerve growth factor is the first nerve food. Is the first nerve food. And what is it? Basically, it is a protein made up of 118 amino acids. Okay. And what is its structure? It has 6 parts, but only the beta subunits. So it has basically 6 parts, but only the beta subunits actually help nerves grow. Okay. It is found in many body tissues. So it is found in many body tissues. Now what is its main jobs here? Helps baby neurons grow. Okay. Obviously, what will the neurotrophins do? Helps the baby neurons grow. Especially the sympathetic neurons. That is why it is called as sympathetic nerve growth factor. Okay. Like pain sensing neurons, cholinergic neurons in brain, like that. Now, it is also used in medicine. That is what we were talking about. NGF, which is nerve growth factor, from the snake venom and mouse glands, they treats sympathetic nerve problems. So, these both help in treating the sympathetic nerve problems. Okay. In the future hope, what we were actually hoping about is it might help the Alzheimer's or aging nerve damage problems, spinal cord injury, all of these conditions. Okay. So these are about the nerve growth factors